Welcome. Today we're diving into a really critical and I think often overlooked factor in ischemic stroke, the gut microbiome. We're going to be decoding a pivotal 2025 study from BMC Microbiology by Huang and colleagues. It uncovers a pretty direct mechanistic link between the bacteria in our gut and cerebral infarction. And just so you know, the links to the full bibliography are right there in the description below.
So, let's frame our discussion with a couple of compelling clinical questions. First up, could the trillions of bacteria living in a patient's gut actively contribute to their ischemic stroke risk completely separate from the traditional risk factors we're always looking for? And taking that one step further, could specific metabolic byproducts from these very bacteria act as a kind of smoking gun, a measurable biomarker in the blood that points directly to the pathology of a cerebral infarction? These are the exact questions we're going to unpack.
Okay, so let's get right into the evidence. The investigators started by profiling the gut microbiomes of 30 patients who had a cerebral infarction, and then compared them to 30 healthy controls. They were essentially hunting for a distinct microbial fingerprint. And what they found was really interesting. It wasn't about the sheer number of different species, the alpha diversity. That was actually pretty similar between the two groups. The real difference, the crucial difference, was in the overall community structure. So think of it less like the number of players on a team and more about who those players are and how they're interacting on the field. Their analysis pinpointed 72 specific microbial taxa that showed significantly different levels of abundance between the stroke patients and the healthy controls. Now, from this big pool of suspects, the researchers used what's called a random forest model. It's a machine learning technique that's really good at sifting through complex data to find the most important predictive variables. Their goal was to isolate a core microbial signature for stroke. And they nailed it. They were able to distill that signature down to just 22 key bacterial genera. That's a microbial fingerprint with some serious power to distinguish a stroke patient from a healthy individual. And this slide really shows you that clear split in the ecosystems. The guts of the healthy controls were much richer in protective phyla like actinobacteriota. These are guys known for producing anti-inflammatory compounds like butyrate. But in the stroke patients, it's a totally different story. Their microbiomes were depleted of these good guys, which basically creates an environment where opportunistic pathogens can take over and thrive. So, let's follow this thread. A different microbial community is obviously going to have a different metabolic output, right? So the next logical step for the investigators was to hunt for the biochemical breadcrumbs left behind by this microbial imbalance, and they did that by analyzing the patient's blood serum. And yep, they found them. 30 distinct endogenous metabolites were significantly altered in the serum of the stroke patients compared to the controls. But out of those 30 biochemical clues, there was one molecule that really jumped out. It's one we already know has some pretty sinister connections to cardiovascular disease, and it emerged as a key player in this whole story. And that molecule is trimethylamine N-oxide, or TMAO for short. Now, the really important thing to remember here is that our bodies don't just make TMAO on their own. It's actually the final product of a metabolic assembly line that gets its start with specific bacteria in our gut. The whole pathway kicks off with diet, things like choline and carnitine from foods like red meat and eggs. Certain gut bacteria then take those nutrients and metabolize them into something called trimethylamine, or TMA. And then finally, the liver takes that TMA, oxidizes it, and turns it into the prothrombotic molecule TMAO, which then gets released into your circulation. Okay, so this brings us to the core mechanism, the real crux of it all. How does a molecule that's made through this gut-liver axis go on to directly impact the brain's vasculature and actually contribute to a stroke? Well, TMAO acts like a systemic agent of chaos. It is not a passive bystander. It actively promotes macrophage activation, it increases endothelial dysfunction, it makes atherosclerotic plaques more unstable, and, this is critical, it helps a thrombus to form. It's literally pouring fuel on the entire pathological fire of an ischemic stroke. The study really cemented this connection with the direct correlation analysis. The very same bacterial genera that were flourishing in the stroke patients showed a strong positive correlation with their serum TMAO levels. This gives us a direct line from the microbial signature in the gut to the metabolic smoking gun in the blood.
So, this all leads to the most important question. What does this actually mean for us in our clinical practice? How do we take this incredibly complex multi-omics data from the lab bench and actually apply it at the patient's bedside?
All right, here are 3 clinically practical takeaways. First, assess the gut factor. When you're managing a patient with all the traditional stroke risk factors, start thinking about their gut health as a potential risk amplifier. Second, recognize the signature. A state of gut dysbiosis, especially one that favors these TMAO-producing bacteria, should be viewed as a legitimate prothrombotic clinical feature. And third, think modulation. This gut-brain axis is a two-way street. This research really opens the door for future therapeutic strategies that could target the microbiome itself to lower stroke risk.
Now, to help anchor this connection in your mind and maybe even start a conversation with a colleague, here's a little phrase to remember. A stroke of insight, the gut's microbial might. It just perfectly captures the idea that what happens in the gut does not stay in the gut. It has some pretty profound systemic and, as we now see, neurovascular consequences.
So to wrap it all up, this study gives us some really compelling evidence that ischemic stroke is tied to a distinct gut microbiome signature, and is a signature that promotes a prothrombotic state, largely through the production of these pathogenic metabolites like TMAO.
And this leaves us with a pretty clear call to action. It's really time we start expanding our conversation on vascular health. The next time you're discussing stroke prevention with a patient or a colleague, bring up the gut. It might just be the missing piece of the puzzle, a powerful and potentially modifiable new frontier in protecting the brain.
Disclaimer and bibliography in the description.